Okay, so let's talk about B cell response and then T cell response. We're going to have some real nice conversations. We're going to start with B cells, right? I believe last week we had some conversation about antigens and antibodies. So we're going to chat B cells that mediate humoral response. B cells originate in the bone marrow. And they mature in the bone marrow as well. Okay. Once they mature, they see lymph nodes, and spleen. Now, what happens during the maturation of B cells? During the maturation, B cells basically are asked one question, quote unquote. Do they recognize self-antigens? What does that mean? If B cells, cells that, as we will see soon, produce antibodies, if they can react and produce antibodies against body's own antigens, body's own proteins, does that make sense? It is not okay. So, if the answer is yes, then B cells go into apoptosis. If the answer is no, then okay, that's great. So this situation when you need B cells to not get activated by self-antigens, it's called negative selection. And when negative selection fails, you got a whole bunch of autoimmune, autoreactive B cells. So far, does that make sense? All right, let's continue. So how does activation of B cells occur? Imagine that we have two populations of B cells. The green population with the specific B cell receptor, it's a B cell, and we can call this population clone 1. The blue population with the different B cell receptor, which we're going to call clone 2. Now, a little disclaimer. You see these receptors? It's a schematic representation. We will address what is a B cell receptor. We will address it later. Okay, so B cell receptors do not look like that. Okay. Now, imagine that these, and there are multiple clones of B cells. Quite frankly, there are billions of clones of B cells that are different, can be differentiated by the B cell receptors. Okay. Does that make sense? So you have billions of different B cell populations, like different families. Okay. So the next thing that happens in our representation is that antigen appears. Now this antigen, look, this antigen, which clone is going to recognize it and bind it? 2, clone number 2. So that moment when clone number 2 recognizes an antigen, we call it clonal selection. So this point, the B cell clone is selected. Does it make sense? Now, the result of that selection is that B cells proliferate and differentiate. So they divide and become more and more numerous, and they also differentiate. And what they differentiate into? Two very specific populations. One population is called plasma cells. And another population is memory B cells. Okay, plasma cells will produce antibodies. And where antibodies are going to be, they're going to be in the plasma. Okay. Now, look, antibodies are in the liquid component of the blood, plasma. The Greek word for fluid, liquid, is tumor. Therefore, we call B cell response a humoral immunity. Does that make sense so far? Now, what are five different types of antibodies? We're going to start with what I believe is the most boring antibody type, which is immunoglobulin E. And its main responsibility, quite frankly, is allergic responses. Okay. Second type is immunoglobulin D, which serves as the B cell receptor. Okay, now look here, I just want to clear, I feel very uncomfortable drawing things like that, but it's better for to exemplify how it works. You see this? B cell receptors are actually antibodies. And remember last week we drew the antibody with FC portion, like, the fork, the handle of the fork. So this handle of the fork, that's FC receptor, FC portion of an antibody. And that for IgD, the FC portion is stuck in the membrane. So basically the whole difference between IgD and other antibodies, IgD are stuck on the B cells. They are not in the circulation. Am I clear? Third type is IgA. IgA is the dimer, meaning that there are two antibody molecules stitched together, and it's found in the mucosa. The mucosal antibody, mucosa like vaginal or respiratory or oral, and this is the antibody that confers protective immunity to children because it is passed in a breast milk. The third type of antibody, is IgM. IgM is pentamer, meaning it actually consists of 5 individual antibody molecules. It's the first response antibody. Okay. And it can activate properly. Good so far? Now the fifth type is IgG. It's the most common type of antibodies. It's produced second antibody, activates complement, and it can also cross placenta. Makes sense so far. Now I would like to address a couple of issues. First, well, it's not really an issue, but the mechanism. How humans can have, look, billions of different B cell clones. How can we do that? How can we provide such an enormous variety of B cell receptors? And the answer to this question is the process called Vdj Recombination. Now, this concept often is confusing, but it is really not that complicated. So let's take a look on the way the antibodies are formed. Like these details, I'm not going to ask you the minute, intricate details of this process, but I want to explain the mechanism. So take a look at a typical antibody. So this is your FC fragment, right? That's the handle before. And the prongs on the heavy chain they consist of three different colors. Okay? I'm going to join, I'm going to pause for a second. And then the light chain will also consist of different colors. Okay, that's quite pretty, right? I'm going to change the colors of the heavy chain for the reasons you will understand this. Now I want you to focus, look, I want you to focus specifically on the light chain, the FAB fragment. It's a little bit cumbersome, but very. So if you remember, last week we discussed that FAB fragment is the one that binds an antigenic determinant. So FAB fragment is the one that has to be variable. Am I clear? Now, you see those different colors, right? Those different colors represent following factors. Let's talk about the heavy chain first. So in order to produce heavy chain, we have sets of genetic fragments. We're going to give them numbers. So 1, 2, 3, 4. 1, 2, 3, 4. And and 1, 2. And let's say it's going to be 3. Now, in the lecture notes, I give some there are 40 something D fragments, 12 something D fragments, whatnot. You don't need to memorize those numbers, God forbid. Look what we have here. In order to produce that part, the variable part of the heavy chain, you need to combine 3 fragments. 1 from V, 1 from D, and 1 from J. Are you with me? So let's say you're combining 1, 1, 1, 1, and 1. It's one combination. 1, 1, 1, right? You can do, for instance, 2, 4, and 3, which gives you a completely different part of a heavy chain. Does that make sense? You understand? So by combining those different genomic fragments in the process of Vdj recombination, you can generate almost, the incredibly large number of variable FAB parts of the antibody. Does that make sense? Now, to make it even better, look at this. You have a certain set of recombinations for a part of the heavy chain and a different set of recombinations for the light chain. It additionally increases the variability. Does it make sense? That's for Vdj recombination. That's it. Okay, we good so far? Now, let's go back to our B cell response. And we're going to answer a couple of questions. Question number 1. What is the temporal dynamic for cellular responses? And to do this, I'm going to draw a graph. And that is one of the fundamental graphs in B-cell immunology. So on x-axis, there's time. On the y-axis, there's antibody levels. Now, let's take a look how antibody levels change after the initial exposure. So here, you have an exposure. First exposure. Exposure can be vaccine, can be an infection, doesn't matter. So antibodies that are formed first are immunoglobulin M, right? So sometime after first exposure, accumulating enough B cells takes time. You're going to have an increase in IgMs. Okay. Slightly later, you're going to have an increase in IgG. Likely. So when you go and, I don't know if you do, you ever decide to go and get tested for antibodies to coronavirus, if you, presuming that the test is good, if the only antibodies that are detected are IgMs, it means that you have an active infection. If you have IgMs and IgGs, you probably have the tail of infection. And if you have only IgGs, You were exposed, you formed immunity, but you're not infectious at the moment. Does that make sense? Now, let's take a look at what happens when you have the second exposure. Are we going to produce IgG? But look how IgG production is. Does that make sense to you? Let me kind of focus your attention on a couple of aspects here. Aspect number one, you see the time it takes after the first exposure to develop antibodies? Can you see that? After the second exposure, your antibodies hit through the roof almost right after the exposure. Does that make sense to you? What provides you with this? Memory B cells. Look, these memory B cells will produce much more, so secondary response is much more robust when it happens much faster. Does that make sense so far? Sure, yeah, I know. Useless. No, seriously. Okay, good. Is that understood? This is your booster vaccines. This is your second exposure to an infection. God forbid. All right? That makes sense. Now, a few more things about B cell response. One, we need to outline the main mechanisms for antibodies to work. And they are pretty well described in the lecture notes. I'm going to list them with some comments right here to save some space. So first is neutralization. In this case, antibodies target viruses. Antibodies bind to the virus, okay, and prevent it from interacting with the receptor. Have you heard about everybody on internet is now making fun of the name of the antibody that Eli Lilly got emergency use authorization recently on? No, haven't heard? It's bamlanivimab. People make all kinds of jokes that I really cannot relate to because of the different cultural backgrounds. But so basically, Eli Lilly got the approval, just like Regeneron did recently, on the antibodies that will bind coronavirus. And that's exactly what they do. They neutralize coronavirus. The only caveat, it's a great treatment. The only caveat is that they have to be used early. I know that some healthcare providers in New York, New Jersey, and Connecticut, what they do if you are positive with COVID and you have mild symptoms, like just mild respiratory symptoms, they will actually send a nurse to you that will infuse those antibodies. And that reduces the hospitalization, it drops hospitalization rate from 30% of infected to 7%, which is quite a lot. Second mechanism is agglutination. Imagine antibodies binding bacterial cells and clumping them together. So they cannot spread, they cannot infect, and macrophages, for macrophages, it's much easier to destroy those clumped cells. Third mechanism is precipitation. Precipitation is binding of various soluble antigens, usually bacterial toxins. So antibodies will bind toxins, preventing them from doing whatever they usually are. The Miller term, opsonization. Remember when we were talking about complement, we mentioned that complement proteins tend to surround pathogens and make them more attractive for phagocytic cells. Does that make sense? Same here. So essentially, increased phagocytosis. So essentially, complement and antibodies both are capable of opsonizing pathogens. And finally, complement activation. No, that is not something we want to talk about. Okay. Good? Any questions so far? No. Correct. Few more things. First, different types of femoral roots. I'm going to throw them at you with the examples, like why the examples. So for instance, if you get infected, infection, is that an act of nature? Yes, so it's natural. And is your immune system exposed? Like, does it actively interact with antigen? Yes. It's a natural active immunity. A vaccine, on the other hand, while you are still being exposed to an antigen, it's not an act of nature. So we call it artificial active immunity. What if child receives immunoglobulin A via breast milk? Is that natural process? Oh, absolutely. But a child isn't being exposed to the antigen. So it's natural passive immunization. And finally, just like in the example that we described and discussed, there's monoclonal antibodies from Cellular, so intravenous immunoglobulin. That's definitely not natural. And your immune system is not exposed to the antigen per se. Okay. Does that make sense? We're good? Now, before we conclude our conversation on antibodies, I would like to stress this. Look at the mechanism of antibody response. Here, this one. Antibodies don't kill. They don't lyse. All they do is they tag. Good? Any questions? Anyone wants to take a picture of this? And now, we're going to discuss the major histocompatibility complex, a concept that is a little basic. So I'm going to be using acronym. Now look at the acronym. Major histocompatibility complex. Histocompatibility. Tissues being compatible. So we're going to start with all of your nucleated cells. All nucleated cells in your body have a special set of proteins. Okay, this set of proteins. it sticks out and waves a label. You see that brown thing? That's the label. Okay? That's MHC plus 1. Now imagine that I am the nucleated cell, okay, right here, and I stick this label out. And the role of this label is to tell the immune system, I am self, I am not infected, I am not nuclear, I'm fine. Okay, that makes sense? Now, what if, let's say it's a liver cell, okay? What if liver cell is infected? Infected with the virus. Then instead of that brown marker, it will stick out an antigen from the virus. Okay? So being infected, cell will wave a different flag saying, look, I am not fine, I am infected. You have to take care of it. Does that make sense to you? Good? That concept? Now, we have also a special subset of cells called antigen presenting cells. Or APCs. There are 3 types of antigen presenting cells. Dendritic. We usually call them DC. B cells. And macrophages. I use this acronym for macrophage. So let me ask you this. Anyone remembers from AP2 the cell types that are not the... It's okay to say no. No. Red blood cells, platelets, sperm cells. Absolutely every other cell in the human body has a nucleus. So, hearing that, dendritic cells, B cells, macrophages, do they have a nucleus? I just told you there are only 3 types with no nucleus. Red blood cells, platelets, sperm cells. Do these have nucleus? They do. They're not red blood cells, they're not platelets, they're not sperm cells. So they do have nucleus. Since they have nucleus, do they have MHC class 1? Yeah, I didn't say anything about exceptions. So let's say this is a dendritic cell. So they have MHC class 1. And these cells specifically have another MHC molecule, which is called MHC class 2. What's the difference? They have slightly different functional roles that we will discuss in a second. Okay? Does that make sense? So in general, what we call the function of MHC proteins is to present the antigen. Look, if I'm the cell, I present the antigen. If I'm that cell, I present the antigen. Am I clear? Yeah. So we're going to switch gears a little bit and talk about the T cell development. T cells mediate cellular immunity. Okay? And this is how it works. So origin T cells is hormonal. Okay? Now, T cells mature in the thymus. Okay, and look what happens. T cells that originated in the thymus, they split into two distinct populations: CD4 T cells and CD8 T cells. Okay? Once they mature, they migrate, so they seed lymphoid organs, and then in the lymphoid organs, they get exposed to the antigen. So we can say activated also in the lymphoid organs. Does it make sense? Are we clear? Now, we're going to go over probably one of the most important steps in the T cell activation. Look, why do we care that there are two populations of T cells? Because T cell cannot be directly activated by an antigen. Does that make sense? So if I am T cell, look, and I see an antigen, nothing's going to happen to me. I'm not going to get activated. Antigen has to be presented to me. Am I clear? How does that happen? Antigen presentation occurs when antigen presenting cell, for instance, dendritic cell, presents antigen to CD8 and CD4 cells, but it presents antigen to CD4 cell through MHC class 2, and it presents antigen to CD8 cell with MHC class 1. Does that make sense to you? Before we move any further, there are four of you here, okay, and I'm gonna assign you the roles. Right? So Anishka and Grace are CD4 cells, and Kenna and Allison are CD8 cells. Okay. I am a dendritic cell. Are you following? So CD4 cells, I present you the antigen on my MHC plus, my right hand. CD8 cells, Ken and Allison, I present you the same antigen, but on the different arm, on MHC plus 1. Does that make sense? Because you, CD4 cells, you can only recognize MHC class 2. You, CD8 cells, can only recognize MHC class 1. This is called MHC restriction. Okay, we're good? That makes sense? Now, how do you remember that? I have my own mnemonic rule, which is it's always 8. 2 multiplied by 4 is 8. 1 multiplied by 8 is 8. It's always 8. We're good so far? Cool? Okay. So both CD4 and CD8 T cells, they proliferate and differentiate. So, proliferation and differentiation. And they end up being effector cells. So what are the effectors of CD8 cells? 2. Cytotoxic lymphocytes, CTLs, sometimes referred to as T killers, and memory CD8 B cells. Good? What are the effectors of CD4 cells? T helper cells, T regulatory cells, and memory CD4. Are you following me so far? Good? I'm not going to touch on the memory cells. It's kind of obvious, but that's your immunological memory. What's the T regulatory cells? T regulatory cells dampen the immune response. They prevent immune response from going out of control. Does that make sense? Clear? Good? Okay. Now, what can T helper cells do? Now, that's quite spectacular. Look, they have three major functions. One, T helper cells can increase B cell activation. On our explanation of B cell function, we showed that B cells can respond to the antigen directly, right? But here, with T helpers, B cells work much better. Am I clear? T helpers can increase specifically macrophage activation. Okay. So can macrophages destroy infected cells? Yes. But with the help of T helpers, they do much better. And finally, T helpers, they assist in the maturation of cytotoxic lymphocytes. Does that make sense? To better understand the importance of T helper cells for human health, the infection that you definitely know called HIV eliminates CD4 cells. Look here. When you eliminate CD4 cells, your B cells don't get proper activation. Your macrophages are not being properly activated. And cytotoxic lymphocytes are not being properly activated. That makes sense? Got it? So your immune system basically is trapped. Now, so far, any questions? This is really a great example of how different branches of the immune system are linked. So for instance, here you have cellular immunity linked to humoral. Here we have cellular linked to innate. Got that? Okay, so let's talk about cytotoxic lymphocytes and how they kill cells. So imagine you have a cytotoxic lymphocyte and you have an infected cell. And it turns out, as far as I remember, and it's not going to be a part of the question on the exam, I guarantee that. Where is what? I believe that FAS receptor is on the infected cell, and FAS ligand is on the CTL. And again, look, I'm not asking you to remember that stuff. When CTL binds this way to infected cell, look what happens. That cell here goes to apoptosis. Now, another mechanism, possible mechanism, CTL will attack the infected cell by releasing proteins or perforins. And what perforins do, they make a pore in the infected cell. Then another set of proteins, so that's perforin, another set of proteins called granzymes, they get through this pore into the infected cell, and what's going to happen? We'll get into apoptosis. Okay, we're good? That makes sense. You can take, if you want, you can take a chair. Okay. Now there are a couple of things. Well, actually, one thing that I'm not going to put in the board, and I'm not going to talk too much about it. I strongly recommend. I kind of insist on watching screencasts. And that one you may have a bit of a blast listening to me writing about vaccines. I'm throwing a big tantrum. Jokes aside, there's a table in the lecture notes for the different types of vaccines. This is something I'm not going to put on the board, but instead, I'm just going to explain what you need to know, what you can expect from the exam. I can give you an example of a vaccine, and you will have to put it in a specific category. So, for instance, if I tell you that injectable polio vaccine contains fully inactivated poliovirus, that makes sense? You look, okay, fully inactivated. Well, that is inactivated vaccine. Am I clear? If I tell you that yellow fever vaccine contains yellow fever virus that has been weakened and it establishes the infection but doesn't cause disease, you tell me, oh, that's the example of attenuated vaccine. If I tell you that papillomavirus vaccine or hepatitis B vaccine contains individual proteins from papillomavirus or hepatitis B virus, you look at it and say, oh, that is subunit vaccine. If I tell you that the current vaccine that we are trying to produce, vaccine against coronavirus, has coronavirus proteins inserted into a vector adenovirus, you tell me, oh, that's the vector vaccine. And finally, I don't know, if there is a vaccine that is a plasmid containing genes from a particular virus or bacteria, tell me that is DNA vaccine. Usually, vaccine questions do not cause any trouble. Okay? And by the way, this is why I think doing the exam on the WebEx will be much better for you. I pledge that if you have a question, if you want some more details, kind of descriptive details, you can always ask during the exam. Like, can you rephrase? Can you describe the vaccine a little bit more? And I will, without giving away the exact answer. Am I clear? Okay. Still have some space here, so we're going to make use of it. We have two big items in the journal left. One is immunopathologies, and another is HIV. So we're going to start with the concept of hypersensitivity. Since everything is being recorded, I will minimize my drawings, kind of highlighting what you must know. The first type of hypersensitivity is called type 1, which is also called immediate. And this is allergy. So in the immediate hypersensitivity, you are exposed to the allergen. Your immune system produces immunoglobulin E, which then attaches to mast cells. When you get exposed to that same allergen, second time, allergen binds to Ig on the mast cells, and the binding causes degranulation and allergic response. If you ever had an allergy, you know what I'm talking about. On the nose, itchy eyes, eyes and whatnot. Am I clear? Now, type 2 is transfusion reactions. This is the hypersensitivity mediated by immunoglobulin G. And this is where we have a chance to briefly talk about the blood types. So you have, first of all, there are, I believe, something like 200 different blood types, which we're not talking about one specific. O, A, B, and AB. Now those letters, A, B, they represent what we call agglutinogens. Those are antigens on the surface of red blood cells. Now, in the plasma, we also have antibodies that we call agglutins. Type O has anti-A and anti-B. Type A has anti-B. Type B has anti-A. The AB blood group has no agglutins, no antibodies. Does that make sense? Now, when blood is being transfused, I have that little rule, sort of rule. When you think about transfusion, pay close attention to the red blood cells of the donor and the plasma of the recipient. Does anyone know the blood types? If you don't mind, can you share? Mine is O positive. Huh? A negative? A positive. Okay. Forget about positive for now, so AAO. Can you take my red blood cells and transfuse to you? You absolutely can, because my red blood cells have no antigens. Does that make sense? Can we do the other way around? No, because your red blood cells will interact with antibodies in my plasma. They will agglutinate. That will cause hypoxia, obstruction of blood vessels, blah, 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 all that kind of terrible stuff. That makes sense. So essentially, the universal donor is type O. The universal recipient is type AB. So these people can receive any blood. I can receive only type O, but I can transfuse to everyone. Does that make sense? Now, what is this story about positive or negative? It's a separate red blood cell antigen that's called rhesus factor or D antigen. So, for instance, I am O positive. These are my red blood cells. Okay. Nishka, I believe you are A positive. So you're going to have D antigen and A antigen on your red blood cell. Now, Grace is A negative, so she's going to have only A with no D. What's important, and that's actually good that I have such a great example here. What's important is that we don't have preformed antibodies against the antigen. So, what if A positive blood will be transfused into A negative person? Your immune system, A negative immune system, doesn't know the antigen. So your immune system is going to form a response. Does that make sense? The other way around is totally fine. So my O positive blood, can I transfuse my blood to A positive? I can. Can I transfuse it to A negative? I definitely should not. Because then, person will form antibodies. And this is where a story about rhesus conflict comes from. So imagine that you have rhesus negative mom and rhesus positive fetus. They are separated by placenta. During pregnancy, everything's fine, okay? But during birth, placenta detaches, and mom's immune system gets exposed to rhesus positive blood of the child. And mom's immune system forms antibodies against rhesus factor. They are IgG. Are you with me so far? Child's fine because it's gone. During the second pregnancy, my mom has antibodies now. During second pregnancy, these anti-D antibodies will cross placenta because that's what IgG can do, and they will attack the fetus. Fetus will develop a condition known as erythroblastoma fetalis. Pretty bad stuff. How can we prevent rhesus conflict? We can give mother an injection of so-called raw gamma antibodies. Okay? So if you are a female and you are rhesus negative, during pregnancy, you will, nobody's gonna poke into your fetus to determine the blood type. Make sense? If you are negative, you're gonna get ROGAM. What ROGAM does, and it's absolutely genius approach, what ROGAM does, ROGAM finds out in the mom's circulation. And when there is a delivery, and those rhesus positive red blood cells get into mom's circulation, ROGAM antibodies jump on rhesus positive red blood cells from the fetus and completely mask it from the immune system. Does that make sense to you? They kind of cover those red blood cells, and mom's immune system simply does not see them. We're good? Okay. Type 3, hypersensitivity, also mediated by IgG. It produces immune complexes. Now, it's a pretty straightforward, unfortunate autoimmune condition. Look, so your immune system produces antibodies that interact with the proteins in the basement membrane of the epithelium. So those antibodies are sticking around in the basement membrane of the epithelium, and phagocytes, I don't have it anymore, but phagocytes, macrophages, and neutrophils starts to migrate to those antibodies and destroying them together with your basement membrane. Am I clear? If that basement membrane is an epithelium of the skin, that's psoriasis. If that's the basement membrane in the epithelium found in your joints in your synovial membrane, arthritis. Your blood vessels, vasculitis. Your heart, endocarditis. Your kidney, glomerulonephritis. What I just described are symptoms of advanced lupus. Okay? It's a true autoimmune disease. And finally, the fourth type is called DNA. It's mediated, it's a cellular hypersensitivity, mediated by cellular responses. Now, that's actually pretty good. I can use this picture. I don't need to show you. Look what happens. Imagine that you are exposed to tuberculosis, and your immune system, particularly your T helpers, they form, well, your CD4 cells form a whole bunch of memory CD4 cells. You're not sick, you're fine, you have latent tuberculosis, but you also have a pool of memory CD4 cells that can recognize tuberculosis antigen. Does that make sense? You go for TB test, and they inject tuberculosis antigen in your forearm, in your skin. The dendritic cells present that antigen to both CD4 and CD8 cells. These T helper cells help to activate macrophages and cytotoxic lymphocytes. Macrophages and cytotoxic lymphocytes start to attack this antigen in your skin. If you have those memory CD4 cells, you produce a massive response. You have that blister, which means you had TB. Okay? And if you never had TB, you really don't have memory CD4 cells, you have no reaction. Anyone ever did TB test? Raise my hand. You go, get injection, and you go back for reading like 3 days later. It takes time for response. This is why we call it delayed response. Good? Questions? I think you get tested when you get pregnant. I think it's a part of it. And I, since our family was all positive, all positive, never had to deal with it. But I spoke with some of the students who received it, and I believe it's in the third trimester, but it's kind of early in the fourth trimester. So I'm going to squeeze that into this space here. Pretty simple. So what I'm going to squeeze here is grafts. Now, grafts is the same as transplant, okay? There are 4 types of grafts. Autograft, from you to you. Usually it's a skin graft. Okay? Isograft. Isograft is the transplantation from the identical twin. That's important. If you have an identical twin, lure them in the basement, chain them there, keep them well. That's going to be a source of performance. Okay, that's invaluable. Allograft. Between 2 people that are not identical twins. Doesn't matter. Total stranger, your brother, really does not matter. Okay? Xenograft is the transplantation from an animal. We don't do much of it. Mostly cardiac valves from pigs. Works really well. Okay? So, side effects of transplantation. For solid organ transplant, it's going to be a rejection. For bone marrow transplant, it's going to be tough versus easy. So let's deal with a solid organ. What is a rejection? So imagine that I am, look at me, because colors will be involved. I'm a person who requires a transplant. Liver. I need a liver transplant. Okay. So I get liver transplanted, and the liver of the donor, does it have different MHC markers? No. That's me. That's me. That's my MHC. That's my MHC. That's the donor liver. Are they different? They aren't too different. They're both brown, right? Aren't too different. So my immune system, which is used to this, will recognize this as a little bit different, a little bit foreign, and will start to mount an immune response to my new liver. Does that make sense? So I'm going to take immunosuppressive pills. If somebody would try to transplant into me something like this, it's not going to work. This is why when people receive transplants, say there should be a match. It's never 100% match, but it's as close as possible. Got it? Now, what is the graft versus host disease? No, this is me. Now, I need a bone marrow transplant, not a solid organ, a bone marrow. So in order to put new bone marrow in me, you need to destroy the old one. Does that make sense? So I get irradiated, my old bone marrow is functionally gone, but all my tissues are the same, right? That's all my tissues. So doctors put new bone marrow that is slightly different. And this bone marrow starts to produce T cells. Those T cells get out, what they see? They see this around them. The entire body is different. Does that make sense? So these T cells start to mount a response against the entire body. Does that make sense? This is why, again, this is called graft versus host disease. The donor's bone marrow mounts the response against the recipient's tissue. Got it? This is why again, match. So far, we're good. Couple more things. Pictures? Okay, I'm going to get rid of this stuff. Because I just wanted to basically finish the immune part and then take it back. Really, really small portion just to kind of outline so we are on the same page. Immune deficiencies. Acquired and congenital. So acquired, you aren't born with those. Most common examples are cancer or HIV, the virus that we're going to talk about. And congenital immunodeficiencies, there are 3 that I can ask you about on the exam. One is agammaglobulinemia, no B cells. Another is DiGeorge syndrome, no T cells. And the third one is severe combined immunodeficiency, which is no B, no T. So far, we're good. So you should be able basically to tell apart acquired and congenital and know at this level of detailization what are agammaglobulinemia, SCID, and DiGeorge syndrome. Good? Okay. So let's talk. We're going to say a few words about HIV. One of my favorite ones. Well, not really. Not really. Well, I'm kind of jealous because it's overstudied. So it is single-stranded RNA genome, envelope virus, and so on and on. What I really want to take home is the infectious process. Okay. So this is a human cell. This is a nucleus. Okay? And this is the molecule. This is going to be the virus. Okay? I'm not going to draw all those multiple. Well, I can actually. So first, here's your virus. Envelope, capsid, RNA. Okay? First thing, virus has to attach. In order to attach, virus uses two different surface receptors. One receptor is CD4. The virus also has to use a co-receptor, CCR5. There's CCR5 or CXCR4. Now, right there, let me explain the importance of it. CD4, does that sound familiar? Those are T cells, right? So virus infects CD4 T cells. Okay. Some people don't have CCR5. Just genetically, they do have, but it's defective. Some people have defective CCR5. It's immune mode. Question: people who have defective CCR5, can they be infected with HIV? Virus has to have two receptors to have it. People that have defective CCR5, can they be infected? No. There is a small percentage of people that are genetically resistant to most common HIV clade 1. Okay, it is true. People of North European descent, Scandinavian, North German, all this. Okay. So eventually, the virus gets into the cell. It can do mostly by fusion when the membranes fuse, sometimes by endocytosis. Long story short, you have RNA, and the capsid of the virus, you see it loses its envelope. Capsid starts to disintegrate, and eventually RNA gets released into the cytoplasm. Okay. Now, I have a very important question for you. So, does anyone remember what's the name for the first step of gene expression? Double-stranded DNA is used as a template for RNA synthesis. Now, DNA, RNA, protein, so DNA to RNA. What is that? Transcription. Very good. Now, what this virus does, and it's a unique feature that actually people got Nobel Prize for discovering it. RNA serves as a template for the virus to synthesize double-stranded DNA. Does that make sense? And this process is called reverse transcription, and the enzyme that facilitates it is reverse transcriptase. Okay? Good part? Now look what happens next. It's absolutely incredible. So, where is DNA in the cell? In the form of double-stranded DNA. So what virus does, it incorporates its double-stranded DNA into human genome, into human chromosome. You clear? Good? This process, the integration of the viral double-stranded DNA into chromosome, is facilitated by the integrase. And now, this is what makes this virus so outstanding. Look. Now, a person infected with HIV has viral double-stranded DNA, we call it a provirus, in their genome forever. Now, that is applied only to CD4 cells. Does that make sense? But all CD4, well, all infected CD4 cells now have virus. And it's not going to go anywhere. What happens next? Virus stays latent after the initial infection, sometimes for years. Eventually, we still don't fully understand the mechanisms of reactivation, but at some point, the genes that provirus, they get expressed. Gene expression. Okay? Which leads to, I'm going to skip like a lot of not super important stuff. Gene expression, viral proteins are synthesized, and you end up with virus particles that leave the cell. Now, this process, when they leave the cell, they have to mature. Maturation facilitated by the protease, viral protease. Okay. Now, when viruses leave, they destroy CD4 cells. Are you with me? If your CD4 cells are gone, remember, the B cells suck, the macrophages suck, cytotoxic lymphocytes suck. Your immune system sucks. Is that clear? Does this process make sense, more or less? Okay? Good. I'm going to put an arrow here in sort of future course. Some stuff about HIV kind of in a bullet points. Transmission. Most important contributor to transmission is the use of intravenous drugs, sharing needles. Most effective way to prevent it is the needle exchange program. We know that it works. Us, my friends. Vice President of the United States, and he was the governor of Indiana. He runs the program, and HIV rates in Indiana skyrocketed. Second, sexual transmission. There are variations. Most risky is anal sex. Virus can be transmitted from the penile mucosa onto the rectal mucosa. Does that make sense? And vice versa. Both ways, but from penile to rectal is more efficient. Vaginal sex works not, chances are not pretty small. Chances are lower, much lower, but they still exist, and nobody wants to take any chances here, right? Speaking of protection, yes, condoms protect. If you will read, there will be 99% that 1% counts for accidents, like sleep condoms. Okay? Oral sex. So I want to make a really important point. While virus can be transmitted from between reproductive mucosa, vaginal, penile, or between penile and rectal, virus isn't transmitted between vaginal or penile and oral. It's not a mucosal route of transmission. Does that make sense? Were there cases of transmission as a result of oral sex? Yes. Because there were wounds in the mouth. Like bleeding gums and stuff. Saying it's almost funny, if it wouldn't be so sad. There were cases of transmission during kissing. The same exact reason. And I found in one review, they call it a violent kissing, which, if you want, try to imagine. Vertical transmission from mother to child. It doesn't have to. If a pregnant woman is HIV positive, she can receive treatment that prevents the transmission of the virus from mother to fetus. Does that make sense? It's briefly epidemiology. You may find that some statements that it's a race disease. If you would look closer, it's the disease of poverty. In every country, it is a disease of poverty. In the United States, if you adjust for income, the infection rates between Latinos, whites, and African Americans are very, very similar. It's all about the income. It's all about access to healthcare, access to sexual education, access to contraception. Okay. Oh yeah, vaccine. Forget it. Virus changes really easily. You don't have vaccine. People really don't put much hope into it. People work on a vaccine, but all attempts were absolutely unsuccessful. But we do have treatments, and this treatment is quite amazing. So we have inhibitors of entry. We have inhibitors of reverse transcriptase, like a ton. We have now inhibitors of integrins. And we have inhibitors of proteins. We really have like 2 or 3 dozen drugs that are effective against HIV. Do they clear the virus? No. Can they reduce the numbers, the viral copies, to be so low that they are undetectable? Yes. That's really, really important. And what is the life expectancy for people with HIV who take those drugs? Same as for people without. They are really, really superficial. Last bit. The person who lives with HIV, they never received a single drug. They received a combination of drugs. Because if you will put selective pressure on the virus in the form of a drug, virus will evolve and will develop resistance. When there are 2 or 3 drugs in the mix, and this mix is changed every 3 years, virus cannot possibly adapt to so many selective pressures, and that's good. Any questions? Injectable. You can transmit. There were hospital-related cases, accidental needle stick, but it's really not frequent. And now we have treatment. So what happens essentially? If you get exposed, if you have such an exposure, you are put on the prophylaxis. I think it's entry inhibitors and reverse transcriptase inhibitors. And in many cases, it actually prevents the infection.
When a patient has sitting for, because we have like this brain infection, and he's sick, he's nervous, going on, it's going to be a HIV, and he's, got sort of bed because he's been violent and whatnot, and he got out of his room before that, before he got started. Actually, so we have 2 different types of restraints, hard and soft. You bit through the soft ones.
Beat?
Get them off.
That in HIV, people don't die of HIV. People die of secondary infection. So for instance, cryptosporidium infection in us, we would have mild abdominal discomfort, maybe diarrhea for a couple of days. And a person with HIV, if they get cryptosporidium viral, they live with it forever. The immune system cannot get rid of it. That makes sense? It's all secondary infection, fungal included.